I'm a postdoc in Luca Comai's lab at UC Davis, and like this protagonist here, we're hoping to be able to engineer plants rapidly, not just for Mars, but hopefully for the vast majority of environmental changes or whatever traits that we would want, and that's sort of the thrust of what our interests are. I'd also like to put a plug-in for social media. If you want to interact more, have questions, I'll be happy to integrate that with this talk. So briefly, I would just like to go through sort of a general overview of methods that are available right now for how we have generally looked at detecting changes in dosage and structure in chromosomes. So one of the most well-known, well-established technique is obviously karyotyping. This is a panel of multiple insertion lines, and this is B73 for maize. It's a protocol developed by Jim Birchler in Missouri. And as you can tell, it's a beautiful way of looking at it, but it's actually a very difficult technique if you've never used it before. And secondly, there's a technique called comparative genomic hybridization, or CGH, that's used a lot in human panels. And in this technique, you basically put DNA chromosomes from a normal karyotype on a slide, and then you hybridize it competitively with wild type and usually tumor. And you can then plot the changes depending on the competition on the array. And then there's the next generation of this. Instead of using DNA, you can basically spot DNA on a slide. And this is usually called array CGH. Secondly, the sort of questions that as a biologist you would like to know of your extra pieces or pieces that you lost in your chromosomes are what are structural changes. So there are, in this paper from Zhang et al. That looked at chromothripsis, which is a sort of new idea on how chromosomes can evolve, and it's been linked to a lot of cancers, in which you have a single chromosome that's highly rearranged, represented by these lines here. And to do this, you either have to use a custom pipeline, which is what we used for our publication recently, or there are actually recently Smuffin, which is something that I've used. It works really well. It was developed for humans, but it works for most plant species, I would say. And in this pack, the great thing about being here is that you get to listen to new things. And there's this thing called NovoBreak. It's not published, but the source code is available. I'm going to try and use that. So, I mean, there's various ways, and this involves sequencing. So, as you can see, there's many different techniques, and you might have to integrate them and get a specialist or collaborate with someone. What I'm going to tell you is that using whole genome sequencing, you can actually get at both the dosage variation as well as the structural changes. So the way that we do this, it was first introduced by Isabel Henry in the lab. She's a research scientist with Luca. So what we do is just basically sequence individuals that we're interested in, and if you have a draft genome or you have a good assembly, you can then map these reads back to the genome. And what we're simply doing is going across the chromosome and look at coverage. So if you have even coverages through each chromosome, in this diploid example, you essentially get a flat line. And in this example with the primary trisomic, where you have an extra chromosome, you get a change in the number of reads that map to, in this case, chromosome 2. So then you will then see the difference. And this is just the theoretical plot that you would expect, and this is just a real-life example of what we see. So it's actually a really nice way to do this. I would also like to point out that because we're doing this by sequencing, we can also call SNPs and look at SNP ratios. So we can even call haplotypes based on, so we can tell which parent, for instance, this chromosome came from. So to do this, we need high-quality library preps, and we've been using various methods, and one of the more important steps is this library prep protocol. And this is just a quick run through of what we've been doing. There's some differences between what high-seq machine you're using. We used to do it on the 2000, and there's a little more leeway on the size of the libraries. I actually typically give them a higher size library because it gives us more power when you're calling structural variation. But with the 3000s, 4000s, if you've never used it, they have this like you have to be at 300 or we'll not sequence it for you. So what we do, we start with about 500 nanograms of good quality DNA. And then we have to share it down to 300. And we use Covaris, so it's just a 96-well plate, very easy to use. And what I do is actually just the PCR-free protocol. And you really don't need much DNA once you go into the high-seq. But I would say the most important part, actually, when you're doing PCR-free is this site selection at the end of library prep after we pool. It's important to, for the 4000 at least, it's important to actually concentrate. So what we do, you just elute it at a lower volume using MPR. So that's a very easy way to do it. So this will be impressed soon by our protocol. And as I mentioned, using the same data that we get, we can, this is actually a shattered chromothripsis chromosome from a population of aneuploids that we've just published on. This is chromosome 1, 2, 3, 4. Arabidopsis have 5 chromosomes. And so we can basically get this sort of data from just one reaction, which is nice. And this is just to show you that it's not applicable to just Arabidopsis. We can also do it in Suecica. Isabel has done this with a dosage variant population that's mutagenized by gamma radiation in poplar. And we can also do it in tomato. I will be showing you data on potato in a bit. So next, in this last section, I'll just give you sort of a real-life example of how and why we're doing this. And this is the main thrust of the research that I'm doing, which is haploid induction through uniparental genome elimination. And it's a really cool system, and I sort of call it the next generation of plant breeding because it allows you to manipulate the genome in ways that you wouldn't be able to otherwise. So why are haploids useful? If you are a breeder and you have a diploid, in this case the example is a diploid, you would typically have to homozygose this individual before you get true breeding populations. However, if you have a haploid induction system in place, you can essentially generate haploids from a single individual, get all the variation in one, and when you make what we call double haploids, you essentially generate a population that is true breeding in one step. And obviously, there are many ways of generating haploids. It's really well reviewed here if you really want to learn everything about haploid induction. There's in vitro methods and in vivo methods, but I'm going to focus on what we do, which is these intra-species or interspecies crosses that are able to generate haploid induction. So when you make an intra-species cross, or interspecies cross, this sort of magical thing happens called genome elimination. And what is illustrated here is that when you have, in this case, an egg cell from the mom, a sperm cell from the dad, typically you make a zygote and you regenerate the chromosome numbers that you would for that organism. But in the case of elimination, one of the parents basically, in this kind of mysterious way, you lose the chromosomes from one parent and end up with haploids. I should mention that it's not clear in this figure, the directional cross does not matter. If you're a haploid inducer, you're always a haploid inducer. And in the case of potatoes, there is actually a natural haploid induction system in place where it's a diploid variety of Solanum tuberosum, which used to be called a different species, but now it's considered the same species. We call them Phorea. And then this is the cultivated tetraploid potato that we generally eat. So we make this cross. The haploid inducer chromosome basically is lost, and you end up with tuberosum haploids. But the haploid is actually still in the diploid state. And there are evidence in which these diploids can then be further reduced to make true haploids, but in potato, we call these monoploids, just for nomenclature. And obviously, this is nice, but then you have to find a haploinducer, and the breakthrough for haploinduction technology came from the work from my previous advisor, Simon Chan and Ravi, where we can essentially make any plant a haploinducer by altering the CNH3 protein. CNH3s are the protein that are found on centromeres and are essential for cell division. And so if you have a knockout of CNH3, you can put in altered versions of CNH3. It can be from a different species or just point mutations. We have work that has been done. And when you cross it with a wild type, you end up with zygote, and elimination ensues. However, it's not just haploids that we get, but we also, a third of the population actually consists of diploids, which are normal. And we also see genome instability, which actually may be related to haploid induction, and we actually see a vast number of aneuploids as well. So a third of the population are aneuploid. And so we went in and looked at the aneuploids and by the dosage plot that I just showed you earlier. But in this case, I also include the SNP plot. We know that it's diploid because one of the haploinduced parent is in the Columbia background. It's just a different ecotype in Arabidopsis, and we cross it to Landsberg, so we can track which chromosome. So if you're a true diploid, you get 50% from each parent, but we're only tracking Columbia in this case. So the biggest class of aneuploids that came out of this obviously were the primary trisomics, shown here for chromosome 3. And you can see the shift of dosage from 2 to 3 copy, and we also see the corresponding change of Columbia DNA that goes from 50% to 66%. And the second class of aneuploids that we identify are what I'm calling truncated aneuploids, in which the chromosome that is the aneuploid chromosome has 1 to 2 breaks. And the third class that I actually showed you earlier was what we call shattered, and these are the chromosomes that have undergone chromothripsis, and you get these crazy changes up and downs in the dosage. So this is all well and good. We tested this individual for X62, and as I mentioned earlier, cytogenetics is one good way to confirm the existence. We have genetic evidence, but it's nice to see it on an actual site. So we did meiotic spreads and did chromosome painting on this individual, and we painted the left arm of chromosome 1 red, right arm of chromosome 1 yellow. I just want to mention there's a little blip of chromosome 4 here, and the reason why this is done this way is because in the haploinducer there's a reciprocal translocation as a result of transgene integration. So there's an exchange that's not present in the wild type. And so from our chromosome DNA fish cytogenetic, we can confirm the fact that the shattered chromosomes actually do exist and do not pair during meiosis with the parental chromosome 1s or 4. I'm just going to show you real quick the correspondence of our chromosome painting with these Circos sort of rearrangement plot. It's actually a really nice correspondence. We were able to create 9 contigs from this chromosome, and I think these match up pretty well with the pattern that we see from chromosome painting. So in the last few minutes, I'm going to spend a little bit of time of what we're doing as using haploinduction and what we think is useful for sort of thinking about combining genome engineering and haploinduction. So I did this experiment where we have homozygous reporter gene. In this case, we're using GUS staining, so it turns the plant blue when you expose it to a substrate. And the haploinducer line has this reporter. We cross it to wild type that does not. And so you end up with, this wild type also has a recessive gene for glabbers that makes plant that don't have trichome, so we can actually tell haploids. And if you look for plants that were all blue, they all had trichomes. And the haploids, most of the time, are all white. But what's interesting are these classes of haploids that have smooth leaves but blue roots, suggesting that there's actually leftover DNA from the haploinducer. We already know that from our sequencing analysis, but it's just sort of a nice confirmation. We also have this weird guy that's smooth leaves, but it's chimeric, that has like splotchy blue from being chimeric. So what are the aneuploid types in haploids? This was sort of buried in the paper, but we actually find disomic haploids. So these individuals have 6 chromosomes. And you can see because of 0 Columbia, suddenly it goes from 0 to 50 for the whole chromosome 4. We also have truncated haploids with truncated chromosomes. So in this case, this is the centromere chromosome 1, and you can see there's a little mini chromosome. So these are really cool because essentially these are introgression lines that carry foreign DNA without essentially any linkage drag. So as a if you work in the mini chromosome field, this is actually pretty exciting. And lastly, we also could see shattered haploids. These are really rare, so I only have one example. So these mini chromosomes were actually first described in a paper in 2011 when they were making lots of haploids, seen as these SNPs that come from Colombia. The parent they used for wild type was different. That's why it's a NFA8 SQ8. But we can go in and actually reconstruct the mini chromosome, and what I found was that these ends that you see here are actually joined together, and it makes a circular mini chromosome. This is confirmed by cytology. This is meiotic spreads of the mini chromosome. You can actually see very clearly here it makes a little ring. What's cool about these chromosomes is that we have now taken it to the F6, and they seem to be pretty stable. So we think that we can actually use this for further improvements. So how does this relate to potato haploinduction? So there have been actually a long history and precedents for haploinducer chromosomes in potatoes. So potatoes, the Freya varieties are shown here. They're usually called IVPs. And there's a bunch of papers by this group in the UK where they actually said there were residual genomes from the haploinducer in the resulting haploids. And it was also reported, so these papers use the same population, and these people repeated experiment with a different IVP 35 variety. So I think it's sort of exciting times. We just are going to start analyzing haploids that we're going to make in-house with our own haploinducer lines using this protocol and using our technique to hopefully see if that actually exists in potatoes. And just as a quick last slide, just to show you with what we can do with available data. The draft genome for potato was a double monoploid that was closely related to the Freas. But so I just made a plot of all the parental lines. This is the tetraploid potato. And what we can actually see already is that there are structural changes in dosage when you map it to the genome. So we're going to slowly try and tease this out, and we actually have these extra additions in the tetraploid potato as well. So just a quick rundown of what I've told you today, that sequencing can be really useful when you're trying to when you're first working on something, a plant, and you have a draft genome. It's really nice to be able to see what you have. You don't have to use multiple techniques if you just use this method. And genome elimination can create heritable genic and karyotypic novelty that we can use. And lastly, I think that we should be able to leverage these techniques to then, in the future, combine it with careful genomic analysis and hopefully be useful for plant breeding. So with that, I'd like to thank the funding agencies. I was with Simon Chan when I was first in Davis, now with Luca's lab here. We have collaborators within UC Davis with the International Potato Center in Peru for the potato project. And these are the other collaborators that worked on the project. So thank you. I like taking the questions.